Hi, welcome to the last chapter of the book that we will be covering in Biology 112, and it is about gene expression, or how is the code that's written by the order of our DNA molecules, how is it read by our body? How are the products of these recipes actually made? The process was first described by Francis Crick. He was one of the two guys that published the paper on the 3D shape of DNA. And surely, from understanding the structure of DNA being two complementary strands that are anti-parallel to one another and that are held by hydrogen bonds, once the structure was clear, the function of DNA, and that's DNA replication, and these processes of DNA expression that we will be studying, they became pretty clear because structure is related to function. This is called central dogma of microbiology, and it states that information, genetic information, flows from DNA to RNA to protein. And the process of information flow from DNA to RNA is called transcription, whereas information flow from RNA to protein is called translation. So every organism that is alive and that we have ever known of to be alive has the same flow, is they have DNA, which then predicts what RNA is going to be, which then becomes the final instructions for making a protein. There are retroviruses that violate this order. Retro meaning like they go backwards, where it's RNA to DNA first. But as we've discussed in chapter 1, viruses are not considered to be alive, so this technically is not a violation for the living organisms. This slide graphically represents this process. It's showing you here that both prokaryotes and eukaryotes have DNA double helix that looks very similar in both species. The order of nucleotides, of course, is different, but the structure is the same. Then we're zooming in here on one strand of the DNA double helix. And this is a simplified model of that molecule. This DNA strand that we're going to call the template strand, because that's what we're going to use to go through the process of reading the DNA, it will be transcribed into an RNA molecule, which will be translated into a protein. So the way I remember the difference between transcription and translation is the fact that when I think of the word transcribe, I think of a scribe. And when I think of a scribe, I think of a Middle Ages monk sitting in the dark place somewhere with a candle, copying the Bible by hand, because that's a lot of what they did. And he is a scribe, so he is copying something. When you're copying something, you are rewriting it in the same language, but you are, using your own handwriting. So going from DNA to RNA, you're still in the language of nucleotides, and you're just simply switching from DNA nucleotides to RNA nucleotides. And that's like having a different handwriting, but staying in the same language. So you're transcribing like the monk in some dark place with a candle. However, information flow from RNA to protein is going to a completely different language, right? It is going from the language of nucleotides to a language of proteins, which is amino acids. And that is translation. So, a little bit more specifically, reading DNA code and using it to build a product, usually a protein, can be divided into two steps: transcription, DNA-directed synthesis of RNA. And you only use one strand of the DNA. So remember, DNA double helix has two strands and they're complementary to one another. For any gene, only one side is considered to be the template or the actual gene, while the other side is just a complementary strand that is not used for this process. Thymine, the nitrogenous base in DNA, is replaced by uracil in RNA. But of course, remember there's another difference between DNA and RNA nucleotides, and that is they have a different sugar, right? DNA has deoxyribose sugar, uracil has ribose sugar. So all DNA nucleotides will be replaced by RNA nucleotides during the process of transcription. Then the RNA that is formed through this process is called messenger RNA, and it will be used to directly synthesize the polypeptides, which are long chains of amino acids. Translation process is the synthesis of polypeptides. It takes place at a ribosome, and it requires several types of RNA. So before we can continue to look at the details of this process, we need to be introduced to some of these RNA types. So all RNA is the same in structure. It's all long chain of RNA nucleotides of various lengths. But when these RNA molecules have different functions, they have a different name. Okay, so these are differing by function, not by structure necessarily, not by the basic structure. Messenger RNA is the RNA that is the in-between information from transcription to translation. There's also ribosomal RNA, which is RNA that is actually part of the structure of a ribosome. We learned about this back in chapter 4, that ribosomes are protein building machines that are made up of a bunch of proteins and RNA molecules inserted in between the proteins. So it's actually part of that structure. And there is transfer RNA, which helps in the translation process, and we will meet it later. There are many other types of RNA. You do not need to know these other types, just to let you know that your cell is full of diverse functions for RNA, which is why it was so hard in chapter 3 to describe directly the function of RNA. One question that often comes up is like, what is the point of this RNA in between step? Why is that even there? And analogy I like to use that helps me understand the kind of the basic reason is, imagine that you have a grandmother who has an old book of recipes that have been passed on in the family for 200 years, very fragile old pages, and grandma is kind of the keeper of this family recipe book. This will be our DNA, or the total information, and it is stored in the nucleus. So because there's only one copy of this for the whole cell, or one copy for the whole family of this recipe book, and it's really fragile, and it has all the recipe how to cook anything in the family, that book is safely kept at grandma's house because grandma has several grandchildren and maybe great-grandchildren. And if everyone was just like taking it from her house, oh, I just need to borrow it, I'll bring it back, then of course it would get lost, ripped, stuff would get spilled on it, etc., and the recipes would not become readable, or you would lose the information from the recipes. So if you want to go to your grandma's house and say, hey, can I see the recipe for your meatballs, then can, grandma will say, yes, but you need to sit here at this table. Here's a clean piece of paper and a pencil or pen, and you can copy this recipe to take home with you. So you would sit at grandma's house where the ancient family book, that's so important for the whole family, is safe and sound. You carefully open to the page that you need, this case for your meatball recipe, and you copy the recipe in your own handwriting on a separate sheet of paper for yourself. And then you can fold that sheet of paper, shove it into your pocket, and go home and use it. And it's a disposable piece of paper. You always know that if you need another copy of the meatball recipe, you can go back to to grandma. But if your cousin, on the same day needs a recipe for apple pie, she or he can go to grandma and the whole recipe book is going to be there. It's not going to be missing because you took it home. So the DNA that has all the information for how to make not only that one cell, but the whole body is like the entire recipe book for how to make all the food that your extended family eats. And the recipes therein are the genes. This mRNA is like the piece of paper onto which you rewrote a specific one recipe that you care about in that moment via your own handwriting and took home. And then when you come home, you use the recipe to make the meatballs, which will be like our product here, the polypeptide or the protein. Transcription is this rewriting the recipe in your own handwriting to take home, and it's a disposable bit of rewritten information. This is the process of reading DNA at a specific point. So you're not reading the entire chromosome. You're reading only a gene of interest. Kind of like you were looking for the meatball recipe, you might be looking for how to make melanin, the pigment that's in your skin, hair, and eyes. In order to copy this gene of interest, this recipe of interest that your cell needs for whatever reason at that moment, you need it, you're going to make it. You need the enzyme RNA polymerase. We already met this enzyme in the previous chapter. Remember, to polymerize is to make a polymer or a long chain of similar subunits. So RNA polymerase is an enzyme that makes RNA polymers. Okay. It starts with the process of initiation. So initiation is like the beginning phase of setup for this process. The synthesis of this messenger RNA does not require a primer because, as you learned in last chapter, RNA polymerase is an enzyme that can start building a polymer of nucleotides from scratch. So even like in the thin air, it can just start building a complementary strand. It requires three things to be put together: promoter, start site, and then eventually a termination site. Okay. So what the heck is a promoter? Let's look at that. Promoter is a region of the DNA. So if you have a long strand of DNA, and part of that DNA is the gene, meaning the actual information for how to make a specific product, the actual recipe for the thing you need, then a section of the DNA polymer that is upstream or before the gene, it is like a landing site for the polymerase enzyme. It's not part of the gene, but it's like a message that is hailing the enzyme, like, come over here, land on this section, so you'll be right before the beginning of the gene. Come land on this. Okay, so it is actually part of DNA on the DNA strand. It's on the same strand as the gene, but it is not part of the gene. It is a kind of like an intro message to the recipe before the gene starts. It is not going to be read and copied for our purposes, but it's very important site for the RNA polymerase, which is the enzyme that's going to be reading and copying the gene to land on that part. So here we see that they've color-coded the DNA double helix in purple and blue to show you that one of the strands is the blue is the template strand, and that's the one we're going to be reading, and the other one is called the coding strand. It's a little bit confusing because that makes it sound like they're both important, but there's a reason to this. We don't need to. I'm not going to ask you about knowing the coding strand, only that you know the term template strand, which is the one that is read. Okay, so on the template strand, the one that is important for us to read is the promoter region, and it is a sequence of nucleotides that is 35 nucleotides before the actual start site of the gene. The only things you need to know about the promoter region is that it is actually a certain part of the DNA strand. It's actually made up of nucleotides. It is on the DNA polymer. And it is the place that RNA polymerase finds, the enzyme finds, in order to begin the transcription process. So when the RNA polymerase binds to the promoter region, it knows where the actual start site is from there of the actual gene. So this is the beginning of the recipe, so to speak. There are several other proteins and things that bind to the area, and now we're ready to go. We're ready to read the gene and create an RNA copy of the gene. So that next process is called elongation, right? It's actually making a polymer of RNA while using the DNA as a template. There's like a bubble that opens, so you have to unzip the DNA double helix to open it up. And in this bubble, we're going to have the RNA polymerase, the enzyme that's going to do the reading and the copying, DNA template, the actual part of the DNA double helix, one of the strands, and one section. This is not reading the whole thing. This is different than the previous chapter. And we're going to have the growing RNA copy that we're making, or the RNA transcript. Okay. After you read the section that the RNA polymerase reads the section of DNA that it's interested in copying, the bubble closes back up and the DNA double helix goes back to the way it was before, and it is ready to be opened and read for another purpose, 5 minutes later. So let's look at what it looks like. This is the DNA double helix. It opens up, so the hydrogen bonds between these nucleotides and these complementary nucleotides had to be broken so it can be unzipped and opened. Template strand is the strand that is going to be read by the RNA polymerase. And just as the RNA polymerase did in the previous chapter, when it sees an C, it puts a RNA G nucleotide. When it sees an A, it puts RNA U nucleotide. G, C, A, U, T, A. And it creates a complementary copy of the gene of interest. So if we are copying meatball recipe, or you're copying the gene for how to create melanin protein, then you have just now created not an identical copy of the gene, you have created a complementary copy of the gene, right? And it is growing and will separate it's just, but you see how it's temporarily connected to the template strand by hydrogen bonds. But as soon as the DNA starts winding back up to its original shape, this RNA piece is going to fall off and be a separate free-floating piece of nucleotides. And we call it the messenger RNA or mRNA. Okay, so elongation is the process of making the messenger RNA from the template strand as a complementary copy. Termination is the ending of this process. It's when the RNA polymerase hits a part of DNA sequence that tells it to stop, disassemble the whole complex of proteins, and for DNA to get rewind. Okay, so it causes information that allows the RNA and DNA hybrid to dissociate. So again, it's going to be reading this template strand until somewhere along the line of the template strand, there's going to be a code that the RNA polymerase enzyme can understand that says, stop, you're done, that's the end of the gene. Okay? And that causes the whole thing to go back to normal, and what you have left is a separate RNA polymer or mRNA that is floating around in the nucleus separately from the DNA. This is called termination. So as you have this separate chain of RNA nucleotides floating in the nucleus that is the complementary copy to a gene, it is almost ready for the next step. But before it can continue on to the translation step, it needs to be modified. There are 3 steps in mRNA modification. First is on the 5' end of the polypeptide, the enzymes in the nucleus add what is called 5' cap. Okay, let's see what that looks like. It's actually an addition of a RNA G nitrogenous base, but it's modified with 3 phosphate groups. So we just call it the 5' G cap. You put it here on the end, and it helps the mRNA from fraying and getting degraded and broken down. Messenger RNA molecules are not super stable, and they have to travel places where they bump into things and they hit stuff maybe on the way or something touches them. It is important to protect the endings, which are the most vulnerable to getting broken or damaged in another way. On the opposite end of the messenger RNA, on the 3' end, poly A tail is added, which is a bunch of RNA nucleotides that have the A nitrogenous base, and they're just added, and it varies how many there are. So now both ends are protected, the G cap on the 5' end, poly A tail on the 3' end. Okay, and then there is a very interesting thing that happens, and that is the removal of non-coding sequences, okay, which is called RNA splicing. So this is the odd thing, is not all of the nucleotides that are in this region are actually important in coding information for how to make our product. Some of it is non-important information, or actually the wrong information. And there is a whole set of enzymes that know how to read this and how to get rid of the non-coding information. Introns stands for intervening sequences, are the non-coding sequences. So section of that mRNA that are not important parts of the recipe you're trying to make. So they're not part of a gene that you're trying to make for the product. they're intervening, like they're in the way. And exons are sequences that are necessary and do need to be kept. Okay, so you need to know that introns are things you're going to throw away, exons are parts of that message that you are going to keep. And spliceosome is a whole collection of enzymes that work together to get this job done. So here what it looks like, you had your original DNA double helix, and you transcribed a whole section, the whole section of it. Okay, so you made a copy, and this is your messenger RNA copy that you made during the transcription period. However, these purple sections of the nucleotides are not important for the product you're going to be making. So they need to be removed. These purple sections are the introns, they're going to be removed, and then the exons, or the important yellow sections, are going to be spliced back together to create the mature messenger RNA. This is the final messenger RNA product that's ready to go to the next step. This process is actually kind of shocking. It was for scientists to figure out that this happens. It seems to only happen in eukaryotes. And it's quite complex how this spliceosome, the collection of enzymes that is in charge of cutting out the introns, how they can detect where the introns are. They make these loops and then they cut them off. You don't need to know the details of this. It's just super cool how this process is happening. And you might ask, what was the point of using all this ATP energy to make a messenger RNA and copy a bunch of stuff that you didn't actually need? Why not just copy it correctly the first time, or why even have those messed up sections? Well, they're actually, we now know they're not messed up sections and they're not completely useless. We know that single primary transcript, so before it gets altered and cut up, it can actually be spliced to make different mature mRNAs. So what that is saying is that the same gene in eukaryotes can actually be made into several different products depending on what introns are cut out and what exons are spliced back in. So it's almost like a recipe with a bunch of optional sections in it that, depending on which ones you keep and which ones you leave out, will give you a different product of the dish you're going to eat. There are several genetic disorders in humans due to mistakes that are made when this process is happening. So estimated 15%. And 35 to 59% of human genes exhibit some form of alternative splicing. So a bunch of these original DNA genes that are in your double helix can be copied, and then depending on what part is cut out, it will give you a different product. So it's almost like reusing the genes to make many different things, and it's pretty clever. So it explains how human genome only has about 25,000 genes, but we actually can make over 80,000 different messenger RNAs, which can become a different product from those 25,000 genes. So complicated, yes. A little bit confusing in the beginning, yes. But genius, yes. So at the end of transcription, G cap is added to the 5' end. Poly A tail is added to the 3' end. Introns are taken out, they're cut out and recycled. Exons are spliced back together. You have your mature messenger RNA, which is the ending of the transcription process entirely. This mature messenger RNA is ready to leave the nucleus through those nuclear pores, go into the cytoplasm where it will be translated. And that is the subject of the next video.